Good morning. I'm sorry we can't get the mic working, so I'll just have to yell, and then I'll go out and find an AV person who can help. So this morning we're extremely lucky to have all day as part of our mini symposium on missing data. And we're kicking it off with a bright and early 8:30 seminar. So thank you for getting up. And we're passing around a sign-up sheet, so it would be great if you could sign in. Anyway, our special visiting scholar for the day is Dr. Jim Neaton, and sitting right here. And Jim comes from University of Minnesota, where he's professor of biostatistics at the School of Public Health. And he's been there for many years. He's also adjunct professor of medicine, infectious diseases, and international medicine. And he holds the title of Distinguished International Professor. His research interests are clinical trials, the design, conduct, and analysis of clinical trials. He's directed statistical coordinating centers for several NIH-sponsored studies, and he's currently the principal investigator of the leadership group for the International Network of Strategic Initiatives in Global HIV Trials, or INSIGHT. He's a fellow of the American Statistical Association and the Society for Clinical Trials. He's past editor of Control Clinical Trials, now called Clinical Trials. He's been recognized by the University of Minnesota with 2 awards, the membership in the Academic Health Center Academy for Excellence in Health Research and the Global Engagement Award. He's a regular member of FDA advisory committees and also of the Cardiovascular and Renal Drugs Advisory Committee. He's served on many data and safety monitoring committees for clinical trials sponsored by NIH, the CDC, and the VA. He's also participated, and this is very important in terms of our topic for the mini symposium today, in the National Academy of Sciences panel on missing data in clinical trials. And so he has a lot of insight from that report and from his own research on missing data. So let's start out.
Wow, thanks very much, Kay.
Thanks, Jim. Thanks for being here.
Yeah.
And how are you going to be okay with?
Yeah,
okay.
I think I put the stool there. I woke up a little weak this morning. I don't know what happened, but if I move toward the stool, somebody will try to catch me, I guess, before it's all over. So it's a real pleasure being here. I feel like in talking about clinical trials, I'm preaching to the choir in a way. And so I was thinking about what to do, and I thought I would focus on some issues that have arisen over the last decade or more in doing international trials. And I intentionally put the title kind of in a very positive way, The Juice is Worth the Squeeze, because I think the future in my mind is going to be more and more global trials. And so I'm going to talk about some issues which come up there, which are real challenges, but I want you to take it as a challenge. And kind of, so many of you look so young here, and I know many of you are students. Your generation will be doing more and more global trials, and challenge you to kind of step up to meet these challenges. So I'm going to talk in that context about the network that Kay mentioned called INSIGHT. And it's an HIV network, although for the last 4 or 5 years, NIH and CDC, because of the scope and the breadth of the investigators in the network, have asked us to kind of work at influenza. I'm not going to talk too much about that these days, but it's a very interesting area as well. The squeeze, I'm going to talk about the challenges first, the juice, why it's worth it, and then summarize. And so INSIGHT, just a very brief background, was funded in 2006, and it evolved through a series of collaborations of trials that were referred to, called ASPREE, SMART, and then SILCAT. And then later, we are now conducting a study called START, a large trial in early treatment. This idea for this international network evolved back in the early to mid-90s, primarily initially with the Medical Research Council in London. And because I realized when I got involved in HIV research, they had many similar ideas about the types of studies that should be done. And so the network is unique, I think, in the sense that we have international coordinating centers around the world And the international coordinating centers have strong statistical data coordination capabilities. Oh, you want to mic me? Okay, I'm sorry.
It's okay.
I'll just stop for a minute. Sorry about that.
No problem. We're sorry about that. Just keep going.
So, I mean, there's the concept of regional centers in clinical trials. Larry and I are sitting on a DSMB for a study called SPRINT, which has several regional centers in the United States. And that's been that model has been used for several years for funding trials funded by NIH in the United States now. But this may be unique in the sense that it includes coordinating centers around the world, and they tend to be data coordinating centers in their own right. So why did we form this network? We had a shared vision, namely we wanted to do large randomized trials, and felt that we could not do them rapidly and cost efficiently just in the United States, or in the case of the Medical Research Council initially in the UK. With the idea that we could faster translate the research globally, we'd get faster answers. And I think probably the group as it evolved was that the working with people that had different ideas about how to approach problems. And I think it's hard to put my finger on it, but that's been a very special part of doing global international trials, and in terms of bringing new ways of thinking about problems to the table. So at the time we formed it, I think many of us, many other people, this far side slide, that the origin came right about the time of the collaboration when we began data, and people just felt that we don't need clinical endpoint trials anymore. And so, and it's true, the way we had been doing them was no longer needed to get drugs rapidly on the market. There were better means now. But there was still the need in our mind for strategic trials that only could be addressed but with long-term clinical outcomes, and that was the way we set up our mission and our collaboration. So it may be of interest to show you what we've done. We have 4 international coordinating centers. Okay. All right. And about 20 what we call site coordinating centers. And just to illustrate how it works, the international centers are in Sydney, the University of New South Wales, in Copenhagen at the University of Copenhagen, and in London at the MRC, and in Washington, D.C. Through the Georgetown University and the VA. Each of these ICCs have associated with them, I'm really talking about, have associated with them many site coordinating centers. And site coordinating centers typically are countries. And these countries, just to illustrate here, for example, Sydney works with Thailand, sites in Argentina, now also in Chile, in Mexico, Singapore, Japan, as well as in Australia. And then these site coordinating centers then work with a collection of sites. So this is just a small number of sites in Thailand. There's actually, in the current study we're doing, I believe, 14 sites in Thailand which are enrolling patients. And so we've set up a distributed network to work in countries around the world through international coordinating centers, but at the same time do everything through common protocols, common data collection schemes, and one database. And so just examples, in Minnesota, where we have responsibilities for working with the ICCs to develop the manuals, regulatory issues, specimen repositories, the randomization schemes, although the randomization itself is carried out through the ICCs, and biostatistical input in terms of design and interim analysis. The ICCs, as I mentioned, all have biostatistic capabilities, as well as local regulatory responsibilities for the different countries that they work with. They actually carry out the randomization and work with the site coordinating centers to do the protocol training. And the site coordinating centers do whatever on-site monitoring is required, translations, interim specimen storage, where specimens are sent to a central repository, in our case, to New Jersey, and many clinical sites that we try to make life as easy as possible for, and kind of focus them on enrolling people, consenting individuals, and following individuals, but they obviously have ethics committee and IRB responsibilities at the local level. So this is a simplification of it, but the notion was that in order to do something really of a global nature, we had to do things in a very distributed manner and kind of share things. And so it was very interesting when we started out. You might imagine, like yourself, you're kind of you're used to doing things as a data coordinating center or a statistical center for many years in certain ways. Well, all of the groups were all interested in doing their thing in a certain way. So basically, we came up with a mantra that we were going to be flexible, we were going to keep it simple, and we were going to have fun. And that nobody was going to interfere with those three things. And as a consequence, there were a lot of compromises that we all made in order to kind of do this together, attempting to kind of use the best practices at these centers everywhere to build the kind of the network. So the trials we've done, just to give you an idea, and I did this kind of, I say patience because, and I put up here not the date when the trial began enrollment, but the date when, approximately at least, when planning began. And if you just compare the date of study closure with the date of planning, you can see an awful lot of time has gone into the planning effort, the enrollment, and the follow-up of these studies. So that when we go to funders, and these are pretty much exclusively, with the exception of one trial called SILCAT, NIH funding, we were asking to enroll lots of people and follow them for a long time in order to get the clinical endpoints that we need. SILCAT was a different story. It was an example of a pharma, I guess you might say, behaving badly in the sense that the company that made a drug called IL-2 announced in November of one year that they were going to stop the trial because the FDA was not going to allow them to get rapid approval based upon CD4 cell count differences. And the community uprising was amazing. And so we convinced SILCAT essentially to kind of turn the trial over to academics and let us run it and got the company out of it totally. It wasn't a pleasant experience, but I refer to it as the Valentine's Day kind of massacre in a sense, because the agreement, after lots of shouting at one another, was carried out on Valentine's Day in 2003. So the squeeze. And so there are a number of issues which arise in international trials that were totally new to me. And I mean, I actually had no appreciation, I think, 5 or 6, well, maybe 7 years ago now, of what a sponsor was. All the funding we had for studies was through NIH, and I just assumed, well, the funder is the sponsor. Well, as a consequence of the European Trials Directive that came out in Europe in early 2000, the NIH determined that because of something called the Anti-Deficiency Act, they could no longer legally sponsor a trial. So I received a communication from NIH saying, we're interested in funding your trial, but we're not going to be the sponsor because of the identification and insurance requirements, and based on this Anti-Deficiency Act, we cannot kind of budget money for non-specific things like paying kind of the cost of care for a patient. So we were asked to take on the responsibility not only of the data coordinating center, but also the sponsor. And this delayed the trial roughly a year, because not only did it require, as you might imagine, a number of discussions with our Board of Regents, and became very good friends with the General Counsel at the University of Minnesota, and kind of all the issues that are associated with sponsorship. But then once they did approve it, there were multiple clinical trial agreements that for the specific study that this involved, called START, that we had to negotiate to get the donated drug. The other thing that was new to me, and probably most universities have them now, is something called an institutional conflict of interest management plan. And because the University of Minnesota holds the patent rights to an HIV drug that was used in the study, the vice president and the general counsel felt there had to be a management plan to ensure that I or people who work with me did not try to make that drug look good. And so we get adverse events, for example, and toss them in the trash can, or things of this sort. So there was a management plan that we've been working under for several years now to kind of make certain that the trial, nothing like that happens. It was rather ridiculous, I thought, because my arguments kind of were on deaf ears, because I had published with my colleagues 2 papers on the drug Islatravir, basically arguing that it shouldn't be used because it was associated with increased risk of cardiovascular disease. And now we were worried about me kind of tossing adverse events in the trash can. Another squeeze, continuing the review. So many countries around the world don't review protocols and even review them on an annual basis the way it's required in the United States by OHRP. And so, for example, we were in the middle of doing a trial in the UK about 10 years ago, and we had continually requested an exemption for the way they did their annual reviews of protocols. Every protocol that your ongoing study has to be reviewed at committees and IRBs every year. And the UK's policy was only to have the chair review it, and if there were issues with the protocol, then they might bring in the full protocol, full IRB or ethics committee. Out of nowhere, OHRP sent a note one day stopping enrollment at all the UK sites that were doing this trial. And this went on for several months, that we could not enroll any patients by the UK sites there. Then we eventually were able to negotiate another peace treaty where they would continue to work on equivalent protections for how these things were done, and allow us to begin enrolling. I don't think this is something I believe has been under discussion now with changes at OHRP, but I actually don't know the status of it. Others here may know about it. But this is recognized as being a major problem around the world in doing international trials. Under the general heading of what's equivalent protections of risk of patients. The second bullet refers to something called a federal-wide assurance, which also I did not know much about before I got involved in international trials, because my university and your university just kind of does that all for you. You have to kind of renew it every so many years. Well, many of the sites in other countries, if they're getting NIH funding for part of their research, also have to have federal-wide assurances. And a situation arose in France a few years ago where the ANRS, a major funding body like the NIH, basically said, Jim, we'll actually cover all the enrollment of the 12 sites in France, and you don't have to ask NIH for any money for the patients we will enroll, but we don't want to have to mess with all the regulatory requirements that NIH imposes on us. And so OHRP came back and basically said, no, just because you are generating random treatment assignments that will dictate how those patients in that trial in France will be treated, they have to kind of go through and do FWAs and all the other regulatory stuff that was required, which they wanted to get out of by spending a lot of money, their own dollars, to do the study. Ultimately, we negotiated with them that the algorithm for randomization was sent to the MRC in the UK, and they did the randomization on their dollars, so there were no NIH dollars used for it, so France didn't have to deal with the FWAs for all their sites. I mean, it's kind of crazy, isn't it? It's when you think about it, that this was a major stumbling block for countries to work together to finance trials. And so, it's just an impediment to kind of getting global support for research questions. Study drug. This is a major problem when you're doing international trials that you need to pay attention to. There's a variety of problems at every step of the way in terms of getting drug imported into different countries. and the same drug may go by different names in the different countries. And just to give you an idea of what has to happen, this is kind of the first step in the importation of HIV drugs. These are licensed drugs to Peru. First, the Secretary of State at the state of Minnesota has to sign off on it. Then the Peruvian government has to sign off on it. I know you can't read this, but this is the signature of the officials in Peru to receive the drug. And ultimately, the Secretary of State, in this case Hillary Clinton, has to sign off on the importation of the drug for the country. And so this process takes several weeks if you're lucky. There's some countries which are notoriously difficult to get drug into, and creates a real problem. In HIV, it's a big issue because you want people not to interrupt their therapy. And we've had some very kind of emergency situations where that turned out to be a potential lapse. The fourth squeeze that I thought I would mention is that was initially a major stumbling block in working with NIH about the international trials, and it ultimately got the leadership of NIAID involved to basically tell the worker bees, you have to start thinking about doing this differently. I can remember being at a meeting where they talked about site monitoring, going out to sites and reviewing source documents against forms, and they had done some accounting, and going to an international site was going to cost about $18,000 on average, and going to a domestic site was $12,000. Now, I skipped over it pretty fast, but we are currently doing a study that's in 35 countries with 225 sites. And so just you can do the arithmetic, whether it's kind of a U.S. domestic site or whether it's a foreign site, it's an awful lot of money if there's a requirement by the NIH to do on-site monitoring at least once a year in all their trials. And so this ultimately led us, and we're in the middle of doing a trial, a cluster randomized trial, in which sites are randomized to get intensive monitoring using a central database, a local kind of monitoring that we give the sites that monitor themselves. Both treatment groups get that, and then one treatment group gets on-site monitoring once a year, and the other group only gets for cause monitoring in the event something major happens that we think we have to visit the site, with the idea of kind of demonstrating that the data are of equal quality and we save a lot of money. Most recently, in India, that you may be aware of, the legislation earlier this year expanded the liability enormously for trial-related injuries and deaths of study participants. And so that there's language in their new legislation that if you're in the placebo group and something bad happens, you have to take care of that person, or if the person dies, their family, for a period of time that another committee will determine. Or if they have an adverse event, or if they happen to be assigned to a treatment that turns out not to have any therapeutic benefit, and something bad happens to them. So as a consequence of this legislation, the trials, we have 2 sites in India that have enrolled 91 patients. We stopped enrollment, and we're playing it by ear along with NIH. Essentially all the trials in India have been stopped by NIH until their government works this out. And so obviously the patients are very interested in continuing their participation, as well as the site investigators, but it's at a political level that, and these kind of things will happen when you do international research. So India is a question mark right now for kind of doing clinical trials generally, not only NIH-sponsored research, but industry-sponsored research. So sometimes it feels more hopeless than it is. And so I guess I want to come back to the challenge. I mean, all these things, we've addressed in some way or another. And I wanted to kind of toss them out just so that it's not easy. I see Kurt walked in. Kurt, I saw one of your little essays, I forget now, a couple weeks ago about all the things coordinating centers have to do. And that's true if we do international trials, do all those, I forget how many pages of things that coordinating centers have to do. In addition, if you're doing international trials, there's some other thinking that you have to, some other issues that you have to think about in terms of actually pulling them off. And so, but I do think this is the future, and I hope you rise to the challenge. So why? Well, the juice. There is juice, and so you get the first thing is collaboration. This was the initial executive committee for our group from many places around the world, and so that the ability to kind of bring together like-minded people and pull something off like this is something that we're very proud of. I'm going to talk about a couple different trials now where I think it's also been very rewarding to be involved. The first trial is a trial of interleukin-2. I mentioned the SILCAT study before that was stopped by the company. That was done in people with very advanced HIV. Another trial called ASPREE was done in people with early HIV disease in terms of their CD4 cell count, and that was sponsored by NIH. The question was whether or not CD4 cell count increases that have been demonstrated in phase 2 studies with IL-2 would manifest themselves in terms of reducing the risk of AIDS or death. And obviously, as I said before, the company was very interested in just getting the drug approved based upon CD4 cell count increases because it didn't require doing a 5 or 6 year trial on clinical outcomes. But when we went into this trial, we knew that IL-2 increased CD4 cell count. We also knew that it was associated with toxicities. You take it in cycles over a 5-day period. So one advantage of it is you don't take it continuously, but you do take it for 5 days continuously. You do this roughly every 2 months or so until you get your CD4 cell counts up, and then we had a maintenance program. And it was unknown at the time of this whether or not the CD4 cell count increases here were really functional in terms of preventing AIDS or death. So the study design was to randomize a little over 4,000 people to IL-2 plus background antiretroviral therapy, which was standard care, or to a control treatment that got the antiretroviral therapy without IL-2. The plan was to kind of do the study over roughly 7 years for 320 primary endpoints. And at the unblinding that we did for this study, I was blinded throughout. And when I saw this graph, I just was feeling very good. So we were able to take a drug which was reasonably toxic and maintain a sizable CD4 cell count difference between the control and the IL-2 arm over several years. And that's the 160 cell on average, the difference. And you can see in terms of less than 300, which is a concern, 6%, 9%, and above 600, 57% versus 36%. So that we were able to affect the change in the surrogate marker that we had designed the study to achieve. When we looked at the endpoint data, however, it was a bummer. So there was absolutely no difference, 150 events versus 165 events between the two groups. And if you look kind of at the control arm in this study and kind of use that epidemiological data to predict what the kind of treatment difference would have been based upon the CD4 cell count difference, we should have seen a 26% reduction in risk. Unfortunately, SILCAT was the same way. This was a study in more advanced patients. It had almost the identical hazard ratio, close to 1, and this put an end to a lot of studies on IL-2, which had been carried out within intramural at NIH for many, many years. And so that I kind of, this was a gratifying analysis in the sense that we got an answer to a question, and not a lot of people are spending their time with this drug anymore. But it's also led to some things which I don't have time to talk about now in terms of understanding, just expanding our knowledge about immunology and how are these CD4 cell count that we increase with IL-2 different than antiretroviral therapy. So that's an area of fruitful research that we're doing right now. A second trial was called SMART. And this study, I think the juice I would start off by saying is that convincing NIAID leadership to do the study. Back in the early 2000s, we went through a cycle with antiretroviral therapy. We got new drugs in the 90s, and those could effectively suppress viral load. And that had a tremendous impact on mortality and morbidity from HIV. However, the drugs were hard to take. They were associated with a lot of toxicities. And so patients began taking drug holidays. And many investigators began doing studies, some small controlled studies, looking at things, for example, if I interrupt therapy, does it have an impact on, say, lipids? Because some of the therapies were associated with lipid elevations, which we thought were associated with an increased risk of myocardial infarction. Others were uncontrolled studies, typically involving 300 or 400 people that would be followed for a year, and the investigators would say no bad things happened in this study, so that this appears to be a safe approach, interrupting the therapy. Of course, there was no power to make that kind of statement, and many of the studies, as I mentioned, were uncontrolled studies, and the trials were relatively small. So the community was very interested in this question, and NIAID leadership, and I can remember having a meeting with them, can you guys do this? And kind of, yes, we can do it. We have this network, and so it was like, do it. That was the conversation. The results of this study immediately led to treatment changes in treatment guidelines, and I'll show you a little bit. They immediately kind of also began to define a new research agenda in HIV, which is, again, would not have happened without having a clinical endpoint trial of a substantial number of people with stored specimens that could be linked with events. It kind of opened up possibilities of research that we just hadn't had in HIV for many, many years. So the design of this study was to take people on or off therapy and randomize them to a strategy that we call drug conservation. And the idea was that only give them the therapy, the antiretroviral therapy, if their CD4 cell count drops below 250. When it's higher, the risk of opportunistic diseases is much lower, so you can interrupt therapy. The control arm, which was to have been standard of care, although I mentioned many, many patients were taking drug holidays on their own, but the control arm here was to suppress the virus, take continuous antiretroviral therapy throughout the course of the study. Surprisingly, when this study was stopped early, there was a substantial difference in AIDS or death, the primary endpoint, favoring the continuous therapy group. So that you can see for each of the components of the primary endpoint here, there were more deaths on the interruption group, more of what we call serious AIDS events, and more non-serious AIDS events that occurred when we interrupted therapy, even though we thought we were doing it at a safe level and reinitiating therapy as soon as the CD4 cell count hit 250. We obviously have learned a great deal more about the epidemiology of opportunistic diseases since then. The surprising finding in this study, which I think has had an impact in kind of changing the research agenda in HIV, was that we had defined what we called a supportive outcome for the Data Monitoring Committee. And this outcome was a composite of major cardiovascular, hepatic, and renal outcomes. And these outcomes, based upon the epidemiological data which were available at the time the study was designed, were thought to be associated, were considered to be risk of taking antiretroviral therapy. And so the advice to the DSMB that we gave them was that you might see an increase in risk of opportunistic diseases during the course of this study, but we'd like you to hold tight unless there's also consistency in the data for these other major cardiovascular, renal, and hepatic outcomes, which are all associated with a much greater risk of death than most of the opportunistic diseases that we call AIDS. Well, as you can see here, the data that when the study was stopped also found that in the group that interrupted therapy, there were more of these serious events. And that was generally true for every type of what we call serious non-AIDS events that occurred that we tracked during this study. And I can remember some of you here I know, and maybe some people in the audience work with the Max cohort at a meeting that John Fair, who used to be one of the leaders of it, kind of was at a major meeting, and he basically said these serious non-AIDS events now have basically just changed the whole way we look at HIV, because now this appears to indicate that these serious non-AIDS events, cardiovascular disease, end-stage renal disease, and cirrhosis, are associated with interrupting therapy, and that with continuous treatment, the risk is lower. And this was the first experimental, really, evidence on this. So we kind of immediately, upon these results, began to kind of go to collaborators, at least in my case, that I'd worked with years ago on cardiovascular disease, and said, this study was stopped after roughly 2 years, and so people were having MIs very rapidly after interrupting therapy. What's going on here? And so Lou Culler at Pittsburgh and Russ Tracy at the University of Vermont, we engaged them. We got some money from NIH to begin exploring inflammatory and coagulation biomarkers to understand whether or not, as a consequence of interrupting therapy, there was a tremendous inflammatory response when the viral load went back up that was causing these events. And so a major other piece of the juice, if you will, was the ability to link the stored specimens that we saved throughout this study now with clinical outcomes, and also to take advantage of the experimental design and measure the biomarkers on treatment and control groups to understand kind of the impact of interruption on inflammatory and other markers. And ultimately, which I won't talk about today or show you anything, is to link findings with DNA with clinical outcomes. So the initial work we did right after the study kind of ended temporarily, unexpectedly, was to look at 3 biomarkers, primarily D-dimer, a coagulation marker, and 2 inflammatory markers, IL-6 and CRP. And as this report indicates, based upon 85 deaths which occurred in the study, striking associations with these markers and increased risk of death. And so this basically initiated for us and for many other HIV investigators a kind of a line of research to understand more about what are the causes of inflammation, how does that impact the coagulation system for people who have HIV. The second thing we did was to take advantage of NHLBI cohorts like MESA and CARDIA, which measured these same markers, and actually measurements were done at the same lab at the University of Vermont, and asked the question, among people with HIV, are these markers elevated? And so what this graph shows is that with a variety of types of covariate adjustment, CRP, IL-6, and D-dimer were strikingly elevated in people with HIV. And when the analysis was done for a subgroup of people that had a suppressed viral load, we found the markers were also 30 to 40% higher than the general population, age, gender matched controls. And so among people taking effective antiretroviral therapy, inflammatory and coagulation markers like this are elevated. And the other thing that we showed with comparison with MESA and other studies is that the risk associated with given biomarker levels seem to be higher among HIV than in the general population observational studies. So you had kind of 2 things potentially putting HIV patients, even on effective therapy, at greater risk of these serious non-AIDS diseases. They had higher levels of these markers, and the higher level of those markers seem to be associated with a greater risk of disease. So that led to, following the initial case control analysis, the ability to kind of run these markers on patients in multiple trials, like this is SMART, ESPRIT, and SILCAT, and just showing that kind of when you take the control arm in these studies, all which were taking effective antiretroviral therapy, about 80% of them had suppressed viral loads, being in the highest quartile of IL-6 or D-dimer was associated with a substantially greater risk of serious non-AIDS, major kind of non-AIDS events or death. The other piece that we were able to do, as I mentioned, was to take advantage of the randomized design. Because of the broad entry criteria in SMART, we were able to look kind of experimentally at what the effects are on these markers when you stop therapy, but also for people who came in off therapy, their randomization essentially in this trial was to start immediately if you're in the viral suppression group, but to defer it if you're in the discontinuation group. Essentially, a study on the timing of antiretroviral therapy, and found that when you kind of stop therapy, the markers went up, particularly D-dimer and IL-6, and when you kind of initiated therapy, the markers went down, suggesting that, again, the combination of this is that offered some explanation of what happened in terms of these serious non-AIDS events in the SMART trial. So there was an editorial, other people have viewed this trial very importantly, and that this was an example that I think changed the way kind of people viewed HIV research at the time. So the last juice, the START trial, which also kind of followed directly from SMART, was that it now became possible to think about a study of the timing of when antiretroviral therapy could be initiated. For 2 decades, roughly, I had been going to meetings and different think tanks about kind of how do we study whether you should start therapy immediately after infection or whether you should wait until the CD4 cell count drops to some level. And the sample size was always very vulnerable in terms of the doing those studies, so they never got off first base. With the idea, potentially, that actually treating HIV may lead to reductions in morbidity, not only from AIDS, but also serious non-AIDS events, the trial looked more promising. And so we've been doing a study called START now for the last few years, where we're planning to enroll around 4,700 people to understand whether treating people initially at high CD4 cell counts above 500 or deferring to 350 is a better strategy. And so this is the design of the trial. It's described in a recent supplement to clinical trials. We actually maybe we're almost fully enrolled right now, and we may expand enrollment by 100 people because of the people that we might lose in India as a consequence of their legislation. We're continuing to follow them, but are concerned that something may happen where we have to stop. There's a number of sub-studies that once this trial was initiated that we're able to get funding from other institutes to look at ancillary studies related to the heart and the lungs, bone mineral density, as well as neurocognitive function. And so at the end of this study, which we predict will be sometime in 2016 or 17, decision makers that develop policies around treatment guidelines will have a host of outcomes to look at for clinically relevant interventions in a diverse study population in 35 countries around the world, very heterogeneous practice settings, and health outcomes that one can make an intelligent judgment as to whether or not therapy should be started immediately or should be deferred. Currently, treatment guidelines in the United States say everybody should be treated. That's largely based upon observational data and expert opinion. And guidelines other places around the world vary.
Do you prescribe what kind of drugs they get?
No, we've left it very broad, and that's one of the complications of the drug distribution that I mentioned. We have actually 17 different drugs. Anytime the Department of Health and Human Services, Dan, puts a drug on the initial therapy preferred list, we work with pharma to get the drug. So the investigator and the patient can kind of choose from a set of drugs which ones they use, both as first-line therapy and second-line therapy during the trial. Those in the deferred arm can also choose from the same set of drugs when their counts drop and they need drugs.
And there's no concern that physicians might prescribe different sets of drugs to those initiating at 500 and those at 350? Or
that definitely was a cause of some concern and discussion, although the development of antiretroviral drugs, since we have so many now, has kind of slowed down, so I think it's less of an issue. There's a drug called Atripla, which is a once-a-day pill that about 70% of the patients that are prescribed treatment may get just for convenience. That's a good question. We did not want to dictate the drugs. We wanted the study to be kind of more broadly applicable to places. And actually, some countries around the world can't use all 17 drugs, so that they only can use the ones that their country has licensed. In most countries, that's many drugs now. And so I think, I'm optimistic. It'll probably be after I retire that sometime in 2016, 17, there'll be another amazing database of specimens and other outcomes that you could link with clinical data to define a future HIV research agenda. And so that much like SMART was a surprise to us, I think START will probably be the same surprising types of results. So what are we going to do next? And so NIH basically says, I think START's going to define the question, and that may be the case. We've been trying to work with a group of investigators to develop a study we call START Survival in Sub-Saharan Africa for the last 2 years, but I don't think that's going to make it in terms of the interest by funders. And the rationale for this trial is that about 800 or 900 patients in START are from Sub-Saharan Africa, and the morbidity there is different than in middle-income countries and in high-income countries. And so we think the question, from many different points of view, needs to be addressed entirely in Sub-Saharan Africa, where the burden of disease is. Right now, many people are looking at interventions to reduce inflammation among people on effective antiretroviral therapy, and we're doing the same thing. I mentioned we're doing influenza research, which takes uniquely kind of the advantage of our network with sites in the northern and southern hemisphere, so we can enroll people with flu all year round. And I guess the message here is that in the case of influenza and in the case of some of the other studies we've done, it's almost like you build it and it becomes so attractive that we've had situations where the funders have come to us. And so we began doing global research at a point in time, even in HIV, before there was a lot of global research being done. And just to repeat, I think this is your future in large part if you choose to work in clinical trials. They lack glamour, they strain our resources and patience, and they protract to excruciating limits the moment of truth. So I put up a list of 5 trials, I think, and so that's kind of almost a career. That's kind of what I've done since the mid-1990s, in large part. So, but if you put the time in, you have a good chance of getting the right answer. And so the moral of the story is the juice is worth the squeeze.